One.
Okay, this is our project on the development and regulatory approval process of drugs. Objective 1, know the steps of the product development pipeline. The steps are product identification, assay development and data analysis, cell choice for transformation, transfection technique choice, purification process choice, scale-up production, clinical trials, formulation, and marketing and sales. This is a picture of how long it takes. Understand the components of research and development of potential products. Research and development, early stages in product development, including discovery of function through small-scale production. Estimate market size. Not only must there be a large enough market, but a substantial source of product to supply for the market. Identifying product sources and creating a comprehensive product development plan. Most biotechnology products originate from nature. The rainforest is a very good source due to antimicrobial activity in plant extracts. Bacteria and fungi are also produce antimicrobials, which kills/slows growth of microorganisms. And antiseptics are antimicrobials used to clean surfaces. Process of identifying molecules to treat a disease. Finding a compound that is active against molecules associated with the disease must be screened, modified, and tested to meet FDA requirements. The screening is an assessment of thousands or millions of potential molecules. Sophisticated instruments and mass spectrometry used to ensure molecular structure, activity, purity, and concentration. High throughput screening, making and testing many batches of similar compounds at once, makes the process a lot more efficient than testing samples individually. Biochip is a type of microarray that holds thousands of samples on a stamp-sized chip. Potential compounds must be modified to be more effective and have fewer side effects. Synthesis in a lab allows for a large enough supply for testing and sales. Organic synthesis means making organic molecules in a lab from simpler pre-existing molecules found in nature. Combinatorial chemistry is the synthesis of larger organic molecules from smaller ones in a lab. As a company evaluates a potential product, they must make the following assessments. Who will use the product and is it needed? Is the market large enough? Does data support that it will work and do what the company claims? Can patent protection be secured? Is it profitable? Safety and efficiency checks. Make sure it gives no sickness, no side effects, no huge costs, no failure of testing, and no patent disputes. This is called a Comprehensive Product Development Plan, CPDP. Objective 3, know the testing and purification involved in formulation. Scientists perform assays and look at pH, particle size, polymorphism, which is the condition of occurring in several different forms, and solubility. Formulation means marketing the same type of medicine in different forms, such as pills, vaccines, or capsules. Types of assays. We have the potency assay, which determines strength of drug dosage. Toxicology assay finds what quantities of the drug would kill someone or hurt someone's body parts. Stability assays determines conditions that affect shelf life. Pharmacokinetic assay shows how drug is metabolized in the body, and pharmacodynamic assay shows biochemical effect of a drug on a body. So the pharmacokinetic is like the development of it in the body, and then this is how it affects like your chemicals and stuff. Know the stages of FDA approval and why they are necessary. Discovery research, lead compound screening. Promising target or lead molecules are identified and tested as candidates for drug development. Lead molecules. Does the drug candidate work in molecule cells? If so, for how long? Is it feasible to produce this new medicine in sufficient amounts or to treat people? What needs to be done to ensure its safety? What characteristics are indicative of its safety? And is it stable over time? These are the stages of the clinical trial. You can see how long it lasts. Preclinical results from in vitro experiments and in vivo animal models to determine the activity of drug under many different conditions before it can be tested in humans. The FDA needs pharmacological profile of drug in living cells, information about toxicity in at least 2 species, and short-term toxicity studies ranging from 2 weeks to a month. Filing an IND with the FDA. If lab decides to do human studies, an investigational new drug application or an IND must be sent to the FDA for approval. The IND must provide preclinical data to justify the testing of the drug in humans, animal pharmacology and toxicology studies, manufacturing information, and clinical protocols and investigator information. Phase 1, examine drug safety and pharmacology and metabolism, determine drug regimen. Only 1 in 2,500 make it to this stage. Phase 2, confirm safety, determine efficacy in humans over the short term, and help set up parameters of phase 3 trials. 100 to 300 patient volunteers who suffer from the illness are recruited for testing. Risks are usually minimal because safety has been established in the third phase. Pivotal in establishing safety and effectiveness of a drug in a large group of volunteers over a long-term period of time. An NDA with the FDA, new drug application. FDA must review all drugs and clear them for marketing, mandated by the Prescription Drug User Fee Act. Law passed in 1992 that allows the FDA to charge drug companies a fee for reviewing their NDA/BLA. BLA is biological license application. Application with information about the manufacturing process, the chemistry, the pharmacology, clinical pharmacology, and medical effects of a biological product. Medical product isolated from a natural source such as a human, animal, or microorganism. Phase 4. Conditional approval after the drug or a treatment has been marketed. Measure the drug's impact on patient subgroup/population, especially children, the elderly, pregnant women. Supply information on as yet undetected adverse outcomes, especially in subgroup populations. Provide additional information on the drug's long-term morbidity and mortality profile. Watch drug use in public. Objective 5, understand what's involved in scale-up. Definition and production checks. The process of increasing the size or volume of the production of a particular product. Cultures are checked for their growth rate, protein production, and monitored to ensure that they have not been contaminated with unwanted bacteria, fungi, or viruses. The expense is enormous. If even one unwanted bacterium or fungal cell entered the culture, the entire batch could be ruined. And the cultures are big, so. Scale-up process. The process of increasing the size or volume of the production of a particular product. Petri plate. Produced cells aren't enough to harvest and sell, so biotechnology companies grow the cells in ever-increasing amounts in larger and larger containers. The first scale-up is to a small volume such as 50 milliliters of broth. As the volume of cells increase, so does the amount of product. If the first scale-up does well, the culture will be scaled up to a 1 or 2 liter spinner flasks, which are types of flasks that commonly use where there is a spinner apparatus inside to keep cells suspended and aerated. Then to 10 liters, then to 100 liters, and so on. The 6th objective, understand what is involved in manufacturing and bringing a product to the market. Fermentation, harvest/recovery, protein purification, formulation, filling/packaging are all in the biomanufacturing process. Downstream manufacturing, recovery, extracting protein from cell culture. Method depends on protein's properties and if it is intracellular or extracellular. Must lyse cells if extracellular, Detergents, SDS, sonication, high-frequency sound waves, mechanical bursting. Recover from broth if extracellular. Centrifuge, filter, supernatant. Eliminating impurities from a sample. Accomplished through column chromatography. Separation of sample by passing it through